genetic testing for structural rearrangement, sometimes referred to as the third wheel of free. So to give you a little bit of an overview, I'd first of all like to give you a bit of an introduction to structural chromosome rearrangements. I'd like to talk a bit about the origins and types of balanced chromosome rearrangements. Move on to PGT-SR in practice before discussing some of the issues surrounding PGT-SR, and then finally some conclusions. So, structural chromosome rearrangements, they're among the major abnormalities accounting for infertility, and the point is here they involve abnormalities of chromosome segments, not of whole chromosomes. They can be balanced, obviously, so if they're balanced then there's no apparent loss or gain of genetic material, and carriers are often not affected. They're not affected provided that the arrangement doesn't disrupt genes in any way, but nonetheless carriers are at increased risk of transmitting infertility, miscarriage, stillbirth, or indeed affected liveborns. Now, they're at risk of having offspring or fetuses with unbalanced translocations, and yes, they can miscarry, yes, they can be stillborn, but of course they can be children with mental and or physical disabilities. So what can we do about it? Well, we can of course do prenatal diagnosis, but that requires an established pregnancy and the termination of any affected fetus. So PGT-SR provides a unique opportunity to maximize the chances of successful implantation, minimize the risk of reproductive wastage, and to select either normal or at least balanced embryos. Now, up until about 2007, we had a pretty good idea of how many samples had been performed. So between 1997 and 2007, there were something like 4,200 cycles performed. More recently, figures are harder to come by, but it's definitely rising, and it's probably over 10,000 by now. What types of balanced rearrangements are there? Well, there can be many, but by and large, they tend to be formed by double-stranded breaks in chromosomes. Those breaks can be anywhere in the genome, but there are breakage hotspots. I won't go into listing all the mechanisms that can lead to structural chromosome rearrangement. You can see them listed there. But usually in a PGT-SR case, we're talking about the carrier parent passing on the abnormality to the child. And by and large, that makes it something like 3 times more likely than a de novo structural rearrangement occurring. These are just some examples of chromosome rearrangement. So reciprocal translocation, you can see in the top left there. A Robertsonian translocation, an end-to-end fusion of 2 acrocentric chromosomes. An inversion here, so in this hypothetical chromosome labeled A to G, the gene order has been rearranged to A, B, C, F, E, D, G. A complex rearrangement, such as the one here, or indeed an insertional translocation. To give you a little bit of an idea of how a reciprocal translocation forms, you can see the chromosomes are just lying on top of one another. They both break and then reform in the wrong way. Now that's fine for the individual involved as long as a gene isn't disrupted, but it can be bad news in meiosis. So here we can see the normal 2 chromosomes labeled N1 and N2, the 2 translocation chromosomes labeled T1 and T2. Now these chromosomes have got to get through meiosis, so they've got to pair and they've got to synapse. Now to do that, they've got to form a pairing cross like this. Now both chromosomes, 2 chromosomes can go to one pole, 2 chromosomes to go to another pole. That's called a 2-2 translocation segregation. But in some of these configurations, that's what can lead to the unbalanced gametes. So we can have different types of segregation. On the right, we have what will end up being a normal one, and on the left, an abnormal one. So on the left, we have what we call an adjacent segregation. So the two chromosomes next to one another segregating to the poles, and on the right, the alternate ones segregating. So you can see then on the left, we end up with daughter cells that have both gains and losses of chromosomal rearrangements, like so. Whereas on the right, you can see top right, we have normal chromosomes, and bottom right, balanced chromosomes. So when we then get into meiosis 2, what then happens is that the chromosomes then go and segregate accordingly. So you can see on the right then balanced or normal, and on the left unbalanced. And these are some of the segregations that we can then see. So I've shown you just 2-2 segregations, so these are the ones at the top, but we can of course get 3-1 segregations, where 3 chromosomes segregate to one pole and one to the other, or indeed a 4-0 segregation shown at the bottom here. So that's reciprocal chromosome translocations. For Robertsonian translocations, then again, you get alternate and adjacent, and in this case 3-0. So adjacent 1 and adjacent 2, and then these are fertilized by normal gametes. You can see you can get normal, balanced carrier, trisomies, monosomies, double trisomies, or double monosomies. Moving on then to inversions, you can see at the top we have a chromosome that is labeled A, B, C, D, E. 2 breaks in the chromosome, the piece inverts, it refuses like so, such as the new configuration is A, D, C, B, E, which is fine until it tries to work its way through meiosis. So instead of forming a pairing cross like a translocation would, then in this case, A has got to pair with A, B with B, C with C, D with D, and E with E. And to do that, the configuration is a loop, like so. Now, if we get a recombination event in that pairing loop, then what will happen as the chromosomes begin to unwind in the first meiotic division, like so, you see the spindles attaching, then it unwraps in a strange configuration such that you get an acentric fragment and indeed a dicentric bridge, like so. So the acentric fragment not having any centromeres will probably disappear, and then we'll get some sort of breakage here that can impede meiosis, again causing fertility problems, but of course leading to deletions in the chromosome like so, such that when it segregates in meiosis 2, and you can see here now we have certain chromosomes with bits missing, like so. So this is the problem. This is how the chromosomes can arise, and some of the people, the PGTSR patients who are coming along to the clinics, are carrying these translocations, and this is usually determined by karyotyping. Now, in terms of how we go about detecting it, then it very much mirrors what we see in the world of PGTA, and we're going to hear from Andrea in a moment about PGTA. So first of all, the biopsies are, as for any form of PGT, can be polar body, blastomere, or trophectoderm. Tends to be trophectoderm these days. Traditionally, we would splat the cell down onto a slide and perform FISH. The FISH was then obviously superseded by whole genome amplification, followed by array CGH, or more recently by NGS. But of course, carrier mapping or indeed haplorhythmesis can detect chromosome translocations. So we'll just go through these in a little bit of detail. Very hard not to give a talk without FISH, even these days for me. So just showing you an example of a FISH experiment here and the design of the probes. So what tends to happen is there would be a centromeric probe, another probe, another couple of probes that was somewhere just below the telomere. It requires a preclinical workup. It requires fixation of cells to the slide. There can be locus-specific probes like here, telomeric probes, subtelomeric probes, or centromeric probes. And this can still be quite good if there are cryptic translocations. So if the translocated piece is right from the very end, it's a very tiny piece, so sometimes FISH can still be useful. Now, this worked very effectively as a means. It wasn't as nearly as controversial as PGTA until microarrays came along. And I should have, of course, I haven't mentioned this in much detail, but I should, of course, mention that STR typing can be used, but this also, like FISH, requires some extensive workup. Along came array CGH. I'm sure most of the audience is familiar with how array CGH works. A whole genome amplification, a labeling of our reference DNA and our embryonic DNA combining together and co-hybridization to a microarray, followed by analysis. And by and large, array CGH has been superseded by next generation sequencing. And to compare the two, then you can see they both work pretty well. But as with PGTA, our NGS gives us a cleaner signal, a bigger dynamic range, and if there's any level of mosaicism in our PGT SR samples, then our NGS can pick it up. Now, array-CGH and NGS can detect both inherited and de novo structural rearrangements, provided they are unbalanced. Our carrier mapping, on the other hand, can detect our imbalance. So that can be as here, for instance. So this green and yellow stripy pattern is indicative of an extra chromosome segment, or if it were black, it would be a missing chromosome segment. But it can, in addition, discriminate between normal and carrier embryos. And I'll return to that in a little while. It's also important to note, so this is a paper from one that Jara gave back in 2015 in Chicago, that it's a very good idea to use array-CGH or NGS because it can also pick up the incidental trisomies at the same time as the ones involved in the translocation. So the trisomies, partial trisomies, the whole trisomies, the whole partial monosomies, the whole monosomies, including those ones involved in the translocation. So it has extra value to use that rather than a FISH-based approach, or indeed an STR-based approach. So I want to just discuss some of the factors. I won't go through all of these, but some of these factors are worth mentioning associated with PGT-SR. So it's very important for genetic counselors to be getting their heads around these segregation patterns so that they can counsel patients about the foreseeable risks involved in their treatment. So as for instance, the probability of an alternate segregation is greater in Robertsonian compared to a reciprocal translocation, something like a quarter versus an eighth, but segregation rates can vary according to sex, terminal breakpoint type of chromosome. Other things that have been suggested that might affect merit segregation with the stage of biopsy, well, there's no great evidence for that. No great evidence for a maternal age effect for segregation, although other chromosomes involved that are being picked up are obviously more likely to be affected in older women. And then there appear to be more normal embryos at later stages, so some evidence of selection, although the jury is a little bit out on that as well. So we know, for instance, in PGTA that the embryos tend to be less aneuploid. Whether the segregation patterns depends which paper you want to read. I want to give a mention here to the concept of an interchromosomal effect. So if you're not familiar with the concept of an interchromosomal effect, or ICE, then essentially what that means is if you have one abnormality already, e.g. caused by an unbalanced translocation, does that make another one, e.g. a whole chromosome aneuploidy, more likely? Well, this has been studied in sperm and in embryos. In sperm, there's no evidence for this for inversions or translocations, except some studies report that there might be an effect if the sperm parameters are already quite poor. In embryo studies, some papers have supported evidence for a interchromosomal effect when there's a Robertsonian translocation, but not for a reciprocal translocation. But it's fair to say that there is still debate and no consensus for the presence of an interchromosomal effect at the time of writing. It's worth giving a mention here to whether we should deselect balanced carrier embryos. So I mentioned a moment ago that carrier mapping, for instance, can detect balanced carrier embryos. So although the translocation may seem balanced, there may be additional or missing nucleotides, so microdeletions or microduplications, perhaps caused by imprecise non-homologous end joining. This can result in a disease phenotype. So for instance, Alagille syndrome is an apparently balanced translocation, but a microdeletion is caused there, and it does lead to this particular pathology. Now, even if there is no phenotype, a balanced translocation carrier, the resultant child, will have the same reproductive problems as the parents. So there is some debate about whether these should be prioritized or whether normal embryos, not carriers, should be prioritized over carrier embryos. Now, as I mentioned, carrier mapping, haplorhythmesis, but also a technique called chromosomal phasing at the base level, or base phasing, can detect carriers. And importantly, this can be done without preclinical workup, like I mentioned for the fish or for the STR typing. So, in conclusion, therefore, PGT-SR is perhaps the least well-known form of PGT, but nonetheless is an effective treatment for large numbers of carrier couples. It also detects de novo segmental gains and losses. Imbalance is caused by an abnormal chromosome segregation, which can lead to implantation failure, to miscarriage, or to affected offspring. It's basically piggybacked on the technical advances that have come about through PGTA, which, as we know, is far more popular. So by and large, the techniques are near identical for detection. The biggest limiting factor, however, in PGTR is the availability for abnormal or at least balanced embryos. The segregation patterns are affected by the type of translocation. So whether it's reciprocal versus Robertsonian, for instance, or for instance, if it's an inversion or a complex translocation, it's dependent on the chromosomes involved, and it has a sex-related effect as well. It's not per se affected by maternal age, but the number of aneuploid embryos from other chromosomes obviously have a maternal age effect. There are various techniques available. FISH can still be used in rare cases, SNP-based techniques like carrier mapping, but also RACE-GH and NGS. Importantly, however, it's very, very crucial that we keep developing and publishing the data sets, because this is constantly a tool for our genetic counselors who, as we know, are at the coalface in counseling the patients here. So that's it from me, and on behalf of Chera, thank you very much.